The cardiovascular system includes the heart, includes the blood vessels, and the blood. Today we are going to deal with blood. So blood is actually, it's a kind of connective tissue. It's a liquid connective tissue. In other words, there are cells, dispersed cells, and there's an extracellular matrix, as we have mentioned before when we dealt with connective tissue. So it's a kind of liquid connective tissue, and it is connected to the interstitial fluid, to many other fluids which are present in the body. Like, you know now about, like, for example, cerebrospinal fluid, which is produced by the blood and then it returns back or drains back to the blood. The lymph, which we are going to study later on after we finish the cardiovascular system. Again, it is produced in the blood and returns back to the blood. The fluid that is, we call it the aqueous humor, remember, which is present in the eye, in the anterior chamber of the eye. And this is produced by the blood and returns back to the blood. So the blood is in connection with many other fluids which are located in the body. That's why it has multiple functions. The first function is transportation of gases, the oxygen, carbon dioxide, transportation of metabolic wastes, like the wastes that are present in cerebrospinal fluid, or the wastes that are present in the aqueous humor, or the wastes that circulate in the body from many other organs, nutrients that are absorbed from the small intestine, for example, and the hormones that are secreted by the endocrine glands. We mentioned previously that endocrine glands, they secrete into the blood, and then the hormone will circulate and affect either other glands or effector organs in the body. So it is for transportation. It also regulates the temperature of the body. So this is by vasodilation and vasoconstriction. So whenever there is, for example, cold weather, the blood vessels that are present in the skin, they will become contracted, and this will reduce the amount of heat loss. While when there is, when it is hot, then the blood vessels will dilate, and so that more exchange of heat can take place. So it plays a very important role in regulation of body temperature. And also it is important for protection from disease and infection, because it contains the immune system, the cells of the immune system, the white blood cells that will attack foreign substances. They will attack bacteria, fungi, viruses, malignant cells, and other cells that are foreign to the body, and this is part of the defense system. So the three main functions here is transportation, regulation of body temperature, and protection. The blood is a little bit thicker than water. That's why it flows slower than water. It is more viscous and adhesive. Its temperature is a little bit higher than the body temperature by half a degree. It's 38 degrees centigrade, and it's a little bit more alkaline. So 7.3, the pH is 7.4, which the neutral pH is 7, and it constitutes about 8% of the total body weight. The volume of the blood is about 5 to 6 liters in the adult male, average male, adult male, and 4 to 5 liters in average adult female, and this difference in the volume is related to the body volume. So it doesn't mean, I mean, per volume it's the same between male and female, but because the total volume of the average male is bigger than the total volume of the average female, so that's why there is a difference in the amount of blood circulating in either of them. 5 to 6 in male liters and 4 to 5 liters in the female. The blood, as I mentioned, it's a kind of liquid connective tissue, which means that there is fluid and there are formed elements. There are cells. Usually we talk about them as formed elements because, in fact, they are not all of them are cells. Most of them are cells, but the others are fragments of cells, but not actual cells. So the blood consists of the plasma, which is the fluid part, and the formed elements, with cells in between quotation marks, because a small volume of them, a small amount of them are in fact, they are not actual cells, but they are fragments of cells. So the plasma constitute 55% of the total volume of blood, while the cells, they constitute 45%. Most of them, 99% of these cells, are red blood cells, and only less than 1% or 1% is white blood cells and platelets, the non-cellular component, or the, let's say, the cell fragment components are called platelets, and we will talk about them in a moment. You can see here when the blood is centrifuged, where it's put in a test tube, in a glass test tube here, and it is spun rapidly. This will result in the fact that the formed elements will descend down, will go down, and so you can see them here. 45% of this volume is red blood cells. Red blood cells contain the hemoglobin, which is a pigment, so that's why it appears red. And the remaining part of the formed elements, which are formed of the white blood cells and platelets, they will form a very thin coat, we call it buffy coat. Then the remaining part, which is transparent, watery part, is the plasma. The plasma, in fact, not only contains water, so it is not actually, it is transparent, but it is not colorless. It's a light yellow in color because it contains other structures, other substances. So it's 90% of it is water, but the other structures are plasma. They are proteins, the plasma proteins. And these plasma proteins, they are created in the liver, and they have several functions in the blood. They are called plasma proteins because they are confined to the bloodstream. So we have three types of these plasma proteins. We have the albumin, which maintains the osmotic pressure of the blood, and these are the most abundant. You can see here the albumins are about 54%, and then we have the globulins. The globulins that, or immunoglobulins, are involved in the production of antibodies, immunoglobulins. That is part of the defense mechanism in the body. These antibodies will attack foreign substances called antigens, and this results in antibody-antigen reaction. So essentially these antibodies, essentially they are proteins, and they are plasma proteins circulating in the blood. And then we have the fibrinogen. Fibrinogen is another kind of protein that will form a meshwork inside the blood, and this will promote clotting of the blood, in addition to the platelets and the fibrinogen, which form fibrin threads, will result in clotting of the blood. So you can see that these plasma proteins have very important functions. As I mentioned, the albumin is most abundant, and it is maintains the osmotic pressure of the blood. Then we have the globulins, which are for the antibodies, and fibrinogen is for the clotting. The other substances that are present in the blood and they circulate in the blood, probably we mentioned that before when we were dealing with the function of the blood. They include gases like CO2 gas. Part of the CO2 is present inside the RBC, but the other part is dissolved in the blood. Not oxygen, but carbon dioxide. So part of the carbon dioxide is dissolved in the plasma, and the remaining part is carried in the hemoglobin of the RBC. Waste products like urea, ammonia, bilirubin, electrolytes, sodium, chloride, and the nutrients that are absorbed from the intestine, the hormones that are produced by the glands, the vitamins. So all these are other substances that are dissolved in the plasma. So the plasma is not completely water. It is 90% of water, and these are the other substances, mainly proteins, different kinds of proteins, and other substances. The formed elements of the blood, again, to remind you that the blood is plasma and formed elements. The formed elements are either red blood cells, we call them erythrocytes, or white blood cells, leukocytes. And these are of two types, either granular or agranular leukocytes, and the platelets, which are the cell fragments, not complete cells. I mean, incomplete cells. They are fragments of cells. The original cells are large, very large cells, multinucleated cells that are present in the bone marrow. And fragments of their cytoplasm will shed and circulate in the blood and form the platelets. The cells that reside in the bone marrow and produce these platelets are called megakaryocytes. Mega because they are very big in size, so they are megakaryocytes. In fact, all these formed elements of the blood, they are produced in the bone marrow, in the red bone marrow. Remember that there is red bone marrow and yellow bone marrow. The yellow bone marrow is fatty material that is present in the central part of the long bones, but the red bone marrow is usually present at the ends of long bones or in the flat bones like the sternum or in the iliac crest. And it is the cells of the red bone marrow are the precursors of the formed elements of blood. Yes?
So once the platelets are in blood, do they serve a purpose, or it's just...
Yes.
Yes.
So, yeah, the platelets which circulate in the blood, as I mentioned earlier, there will be a mesh of fibrin threads. When there is injury to the blood vessel, this will induce a cascade of reactions, and this results in the formation of a meshwork of filaments made of protein. The spaces in between these filaments are going to be plugged by these fragments, by the platelets. So that they are very important in clotting of the blood. Okay. Now, as I said, that all these formed elements of the blood, they are produced in the red bone marrow. And there are two types of white blood cells here, because the white blood cells have several functions. Like, for example, the platelets are for clotting. Red blood cells, they carry the oxygen, so they have limited functions. But the white blood cells, as we will see in a moment, they have several functions. That's why we have different types of white blood cells, not only granular and agranular, but there are subtypes of each because they have multiple functions, as we will see in a moment. Let's start with the red blood cells, or erythrocytes. These erythrocytes, they contain the oxygen-carrying protein, which is the hemoglobin, and this hemoglobin contains a pigment, a red pigment, and this is responsible for the red color of the blood. The half of the weight of the cell, in fact, contains is formed of the hemoglobin. So it's essentially it carries the hemoglobin, and as we will see now, that these cells of red blood cells, they start as cells in terms that they have the cytoplasm, cell membrane, cytoplasm, organelles, and the nucleus. So they begin as cells in the red bone marrow, and as they mature, they will lose the nucleus. They will expel the nucleus, and then they will start circulating in the blood with only few cytoplasmic organelles for a couple of days, and at this time they are called reticulocytes, and then they will lose these organelles as well and continue as red blood cells. So that's why they have a limited lifespan, because they are anucleate. They do not have nucleus, and they do not have organelles in their cytoplasm, so they cannot maintain themselves. So they will wear out and will be destroyed and have a limited lifespan. The reason for that, they have no nucleus in order so that they can carry more and more hemoglobin, and therefore they contain more hemoglobin, and therefore they can carry more and more oxygen. So they are always destroyed, and they are always replaced by the bone marrow. And this is reflected also on the shape of the red blood cell. You can see it looks like a biconcave disc, and this concavity here, it increases the surface area of the cell membrane for the same volume. Okay, so and at the same, so this means that there is more exchange of oxygen between the interstitial fluid and the cytoplasm. Oh, sorry, yes, the cytoplasm which contains the hemoglobin. So it increases the surface area of exchange, and at the same time, the presence of this thin part here in the center of the red blood cell will make it easier for the cell to change its shape and to fold, because these cells, they are about 8 micrometer in diameter, but they pass into tiny, very small, tiny capillaries. Some of these capillaries are even smaller in diameter than the red blood cell. So that's why the red blood cell has to squeeze itself, to fold and squeeze itself while passing through these capillaries. And having a thin central part of the cell will make it easier for these cells to fold. So that's the reason that they, and actually at the beginning, when these red blood cells are formed in the bone marrow, in the red bone marrow, they are not biconcave. They will become biconcave when they lose their nucleus, when they expel their nucleus. So the expulsion of the nucleus will allow this biconcavity, and the biconcavity contributes, as I mentioned, to increased surface area of exchange, and also contributes to the fact that these cells are more flexible in shape and they can squeeze themselves in between or inside the lumen of very small capillaries. The count of red blood cell is about in millions. It's not like the white blood cells, as we will see in a moment. Like for each 700 RBCs, there is one white BC. So their count is in millions. They are 5.4 million per cubic millimeter in the male, 4.8 million per cubic millimeter in the female. And the reduced amount, less amount in the female, is because the females are repeatedly losing red blood cells during the menstrual period. So especially during the reproductive period of the female's life, usually they have less amount of red blood cells in their blood because they are losing them during the menstrual cycle, and that's why it is less. The count is less. As I said, that these cells, they have a lifespan of approximately 120 days, like 4 months, and they are continuously replaced by a rate of 2 million per second from the red bone marrow. This is how the life cycle of RBC, this wear and tear that takes place in the RBC cannot be repaired, as I mentioned, because there's no nucleus and there are no organelles in the cytoplasm. So these are the worn out RBCs are destroyed by macrophages, phagocytic cells, which are present in the liver and in the spleen. We are going to study these macrophages when we deal with the spleen especially. And so they destroy these worn out RBCs, and then the contents of the RBC, or the structure of the RBC, is going to be recycled. Most of the content will be recycled. Like, for example, the iron that is present is going to be reused again. The amino acids are going to be reused again. But the pigment, the pigment part of the hemoglobin is going to be excreted, and this is going to be excreted by the liver in the bile, which is excreted through which is stored and then excreted through the gallbladder. So when there is excessive destruction of red blood cells, the amount of bile will increase in the blood, and this result in the yellow discoloration of the skin, what we call it jaundice, because of excessive increased amount of bilirubin in the blood. One of the reasons is that there is excessive destruction for one reason or another of the RBCs. So there is more hemoglobin and more heme that is going to be destroyed and converted into bile. This is how the hemoglobin molecule looks like. It consists of polypeptide chains, 4 chains of polypeptides. Polypeptides means multiple amino acids connected together, and it contains the heme pigments. So there is one heme pigment for each hemoglobin, for each polypeptide chain. So for one hemoglobin molecule, there are 4 heme pigments, and the heme contains, it looks like a ring, and in the center of the ring there is an iron ion, and it's the oxygen that is connected to the iron. So therefore, one molecule of hemoglobin can bind to 4 molecules of oxygen. The hemoglobin is not only concerned with the exchange of oxygen, with carrying the oxygen, but also carries the carbon dioxide, as we have mentioned previously. But the carbon dioxide does not bind itself to the heme. It binds to the polypeptide chain. Some of the carbon dioxide is transported by the red blood cell. The other part of carbon dioxide, which is the major part actually of the carbon dioxide, is transported, dissolved in the plasma, not inside the red blood cell. But oxygen is always transported by the red blood cell. Now, the white blood, in the red blood cell, I'm sorry for that. So the oxygen is always transported by the red blood cell. Carbon dioxide is transported either by the red blood cell, where it binds to the polypeptide chain, and this is about 23% of that CO2, but most of the CO2 is dissolved in the plasma as bicarbonate. The white blood cells, as I said, that there are 2 types, major types of white blood cells, granular and agranular. And this description is derived from the fact that there are granules in the cytoplasm and in the granular white blood cells, and there are no granules in the agranular white blood cells. So, or at least the granules will not show by conventional stains and by light microscopy. But in the granular white blood cells, they show by conventional stains and under light microscopy. So the granulocytes are 3 types. They are neutrophils, eosinophils, and basophils. And the subdivision, or these names, are derived from their staining properties, the staining properties of the granules in the cytoplasm. The agranulocytes are either monocytes or lymphocytes. So let's start with the neutrophils. The neutrophils is one of the major components of the granulocyte series, and the other names that they are called polymorphonuclear leukocytes. You can see here that they have a nucleus that has multiple segments, and usually 2 or 3 segments, but when they become older in age, they will have more segments, up to 5 segments of nuclei. So these lobes or segments of the nuclei are connected by thin strands, so therefore they have different shapes of nuclei. That's why they are called polymorphonuclear leukocytes, because the number of lobes varies between 2 to 5 and different shapes, so they are polymorphonuclear leukocytes. And they are neutrophils because they contain granules in the cytoplasm, but these do not stain dark. So they are neutral to the pigments or to the stains. They are neutral to the stains. That's why they are called neutrophils. Okay? And these cytoplasmic or granules, they are filled with digestive enzymes, because these cells, the polymorphonuclear leukocytes or neutrophils, they are phagocytic cells, and so they ingest the bacteria, and the bacteria is digested by these digestive enzymes. Also, these digestive enzymes, not only these digestive enzymes that digest bacteria, but these neutrophils also release substances that act like antibodies, killing the bacteria outside the cell, and also they release some oxidants like hydrogen peroxide, for example, which works like a bleach, and also it destroys the bacteria. So they either phagocytize bacteria and digest it, or they kill the bacteria by releasing defensins or releasing strong oxidants. And whenever there is an infection in the body, then the first white blood cells that will reach the site of the infection are the neutrophils, and they are the most abundant of the white blood cells that are present in the body. Then we have the second kind of group of granulocytes, are the eosinophils, and these have, usually they have a bilobed nucleus, 2 or 3, usually 2, and the granules in the cytoplasm, they are large granules, and they stain with acidic dyes, including eosin. So they love the eosin. That's why they are called eosinophils. They are granulocytes because they have large granules that can be seen under the light microscope in the cytoplasm. And these granules, they contain enzymes that are involved in allergic reactions and to attack parasites. That's why you have eosinophilia, which is excessive increased amount of eosinophils in the blood when there is allergic reaction or when there is a parasitic infection, because they are specific for attacking parasites and in allergic reaction. The others are the basophils. The basophils, again, they have large granules in their cytoplasm. These granules, they have affinity to basic dyes. So that's why they love the base, so they are basophils. And you can see that the nucleus is also multi-lobed nucleus, but the nucleus is almost obscured by the large basophilic granules in the cytoplasm. And these granules, they contain substances that intensify inflammatory reaction, like histamine, like heparin. You can see that there are different functions and different white blood cells. So the basophils, eosinophils, and the neutrophils are the 3 types of granulocytes of the white blood cells. The most abundant are the neutrophils. The agranulocytes are of 2 types. We have lymphocytes and monocytes. The lymphocytes, they have a nucleus that is oval or rounded and almost fill the cell. So they have a very thin rim of cytoplasm, which is agranular cytoplasm, and they are very important in immunity. We have 2 types of lymphocytes. We are going to talk about them in more details with the lymphatic system. There are B cells and T cells. So both of them, they are produced in the bone marrow, but the B cells, they are produced in the bone marrow and they mature in the bone marrow. The T cells, they are produced in the bone marrow but mature in the thymus. That's why they are called the T cells. The B cells, they differentiate into plasma cells that produce antibodies, and while the T cells they're alike, then what we call them the NK cells or natural killer cells. They attack the fungi, some bacteria, but they usually attack fungi, viruses, and they also are involved in attacking cancer cells, foreign cells, when there is transplanted organs in the body. But the B cells, they are the main cells that attack the bacteria by differentiating into plasma cells and producing antibodies. The monocytes, the second type of agranular white blood cells, these monocytes, they have very characteristic nucleus, as you can see them here. It is horseshoe-shaped or kidney-shaped nucleus. They are larger cells than the other white blood cells. They are very large cells, and they are phagocytic cells. Usually, a few of them are present circulating in the blood, but usually they leave the blood and will go to the tissue where they will form macrophages. And it is the macrophages that will phagocytize the foreign substances. We have seen an example of these macrophages when we studied the alveolus, for example. There is an alveolar macrophage, and they ingest, for example, bacteria or particulate matter like the dust and smoke. That's why they are called dust cells. So they reside in the tissue. After they start circulating in the blood as monocyte, they leave the blood and reside in the tissue as macrophage and start ingesting. So some of them are present in the lung, some of them are present in the liver. We are going to study them later on. They have a specific name and they are called Kupffer cells. Yet others continue circulating in the blood. The white blood cells are less numerous than RBCs, as I said. Their number is in thousands per millimeter, not millions per millimeter. So 4,000 to 100,000. When they increase in the blood, this is called leukocytosis, like what happens when the body is attacked by microbes, when there is even exercise can cause increase in the number of white blood cells. When the number is reduced, this is called leukopenia, as what happens during radiation or chemotherapy, which will affect the bone marrow and reduce the production of white blood cells in the bone marrow. That's why you have reduced amount of white blood cells circulating in the blood, so you have leukopenia. And as I said, both white and white blood cells, they are produced in the bone marrow. That's why examination of the bone marrow is very important to give an idea about many of the blood diseases. And this is done by either aspirating the bone marrow or using a bone marrow biopsy. Usually we use the iliac crest because this is a flat bone, so it contains red bone marrow. Or sometimes they use the sternum. Again, it is a flat bone and it is easily accessible because it is just under the skin. So the iliac crest and the sternum, they are just under the skin. You can easily access them, and they contain red bone marrow. You don't go to the center of a long bone which contains red bone marrow. The platelets, as I have just mentioned, they are involved in the clotting of the blood. These are the fibrin threads that are formed when the endothelium is destroyed, and then these spaces in between them are clogged with the platelets, and so we have the clots. So they are very important. As I said, that they are produced by megakaryocytes in the red bone marrow, and they are cell fragments. They are not cells by themselves. This is how to obtain a sample of blood by venipuncture. We use a vein here in front of the elbow joint. This is called the median cubital vein, just under the skin. This is where you can sample the blood. And this is very interesting topic that if you want to read about, it's called blood doping, where you increase the number of red blood cells in the blood, so this will increase the oxygen carrying capacity of the blood, but at the same time it increases the blood viscosity and results in problems in clotting of the blood and obstruction of the blood vessels, which will result in tissue destruction. Blood doping is used by athletes, but now it is banned. But the idea is that you increase the number of red blood cells to increase the oxygen carrying capacity of the blood.